it's my great pleasure to welcome Dr. Renata Jakowska from the University of Cardiff. Initially did PhD and BSc and MSc at the University of Warsaw, if that's correct.
PhD in Germany, but
the PhD was in
yes.
Germany, and then some postdoctoral stuff in Germany, in what's it now? Bremen, and then Stuttgart. Stuttgart, which is obviously where England's great victory was yesterday, so there's good omens all around. And then to Heidelberg to work in COPD in the industry before going to Cardiff in 2019 as a senior lecturer, where she's been working on the epigenetics of COPD and was recently awarded a very prestigious Future Leaders Fellowship from the UK. So it's my great pleasure to invite you here to tell us about your work and about epigenetics. Thank you very much.
Thank you. It's lovely to be here. So thank you so much for coming here and for inviting me to share what we do. So hopefully in this audience, I don't have to excite you about the lung, which is very rare for epigenetics conferences, and I have to explain people why lung. So it's quite a nice change of scenario. But I try to excite you a little bit about epigenetics because I'm essentially an epigeneticist. So that's how I trained, and that's why my heart lies. And I think the whole point, at least how I started, was when I was a PhD student. I just find it absolutely amazing that all our cells have the same DNA, and yet you can create such beautiful variety of morphology. So if you think we all start as a single cell, the zygote, right? And already in the newborn baby, you have about, I checked the number recently, they say 1.5 trillion cells. And if you think about how we grow, develop, age, or disease, there's this impossible amount of morphology, specialization, function can go into. So of course, fundamentally, how can a single genome give rise to all these different phenotypes? And if you think about lung, I think because we now know that despite this one genome, we have different epigenomes in those cells, and that partially drives this functional differentiation and specialization. And if you look specifically, let me make this up see online as well. And of course, we now know that this kind of epigenetic information is encoded essentially in a pattern of chemical modifications to either the DNA, like for instance, the DNA methylation, that's what I'm going to talk about today, or post-translational modifications to the histone tails. And essentially what they do is they, of course, regulate the accessibility of chromatin and thereby transcriptional programs of the cell. And therefore, of course, this is not surprising that epigenetic kind of thinks that epigenetic is, of course, very important in controlling the cell fate and function. But I think in the lung, there's another thing which is very interesting, and this is, of course, how I got into the lung being an epigenetic, because epigenetic mediates or is the interface between the environment and the cellular phenotype, right? And if you think about the lung, that means that if you expose the lung to all varieties of exposures like cigarette smoke or others, you're going to essentially change the epigenome of those cells, and that has been, of course, widely documented. So you can think about epigenetics as well as a cellular memory of the past exposures. And of course, this is not surprising due to the central role of the epigenetics, that epigenetic alterations and regulators are, of course, key drivers of disease. So very simplistic thinking, if we can find or decipher what the cell has been through, and maybe we can also think about remodeling these epigenetic alterations, and that's another way to think about reverting disease phenotypes. So that's basically where I come from the kind of basic thinking. And then what we are interested in the group, from the very basic fundamental process, is how that epigenetic or epigenetic processes drive cellular identity and repair. How do the cells know that they have to differentiate and what they differentiate into? What happens, or how do epigenetic mechanisms respond to a variety of environmental insults? So, for instance, cigarette smoke. And if, of course, more translational, what goes wrong at the onset of respiratory diseases. Model systems, I'm going to talk today about COPD, because that's where we've done some work. But with the fellowship, we will be moving into lung fibrosis, and hence, of course, I'm also here trying to build a bit of a network and collaboration, because I know very little about lung fibrosis, so that's going to be quite a steep learning curve. But what is, again, from an epigenetic point of view, quite fascinating, is that very simplistically looking, you could imagine COPD or emphysema as not enough repair model, while your fibrosis is too much repair model, if you want. And again, how do the cells know what to do? How do the cells know in which direction to go? So essentially, we are asking three kinds of questions. First, what are the epigenetic changes associated with disease development? And potentially, of course, that could give us a disease signature or in the future, potential for patient stratification. What are the key disease phenotypes? And we are very interested in specific cells that drive them, looking a bit more at molecular mechanism because, as I said, my background is in biochemistry and mechanism, so we want to understand the mechanism. And of course, essentially, could we in the future use some of the epigenetic regulators as potential new candidate targets? This is basically what we do. So we work with human lung samples, and I'm going to present to you today one protocol, which I hope will be very useful for the community, which is cryopreservation. We are developing low input epigenomics methods, so mainly DNA methylation, but also we've been now working quite a lot on trying to identify methods for looking at histone modifications from limited number of cells. And we do a little bit of a single cell stuff as well. We built 3D cellular models based on primary cells. And recently we've been very excited because we also managed to establish tools for precise editing of epigenetic states. So we can now change, for instance, a DNA methylation pattern with an enhancer of a promoter in a specific. Not yet as good as we would like to, but it's on the go. And recently we've been developing collaborations to look at guinea pig models for lung diseases. Changing gears, I don't have to introduce the COPD too much here, but I just wanted to bring in the room a little bit of numbers. We know it's very prevalent. We know that it costs a lot, the UK economy, and we do not have effective drugs despite basically decades of research done on COPD. And we know that one of the key phenotypes in the COPD, I'm going to look from the repair point of view, so I'm going to forget the airways and airway limitation for a moment. I'm going to focus on the alveolar space and on emphysema. We know that there is somehow a deficient repair or lack of repair in the alveolar stem cells and also in the niche which is there. So in a way, for us, it's a model to try to understand better why these cells do not repair with an epigenetic lens. So essentially, the first question we ask is which epigenetic changes are associated with emphysema development? And the approach is fairly simple. So we wanted to start with lung tissue samples. So we wanted to start with the human, because we don't really have reference maps for DNA methylation at high resolution from purified cells. We purified fibroblast and alveolar type 2 cells. We also did basal cells, but I skipped them for this talk. And then we did low input whole genome disordered sequencing, which is an unbiased high resolution way to look at the whole methylation landscape of the cells, and accompanied that with bulk RNA-seq to identify changes and phenotypes. Of course, the data integration to identify regulators. And then we are, in a way, at the validation stage, trying to understand better what could be the regulators of the disease phenotypes. And I'm going to show you some of the data. So first trouble came, of course, with tissue. You think getting tissue is difficult? Yes, it is. But even if you get tissue, you start running into troubles. It comes at 5 in the afternoon. What do we do? Or so forth. So what we've spent some time doing, and I'm going to summarize it in a couple of slides, we have managed to develop a cryopreservation protocol for pieces of tissue. And the biggest advantage of it, so essentially what we do, you see it in this little cartoon, we get the lung tissue, we take a piece for HNT so that we can evaluate the quality and also keep a block for validation. The rest of the tissue we put into small pieces, put it in a cryopreservation solution in tubes, and tissue stays viable for years. So we can go back to the tissue, isolate cells, and do our profiling when we are already certain that the tissue quality is what we want it to be, or when diagnosis is not yet certain, we can wait until the diagnosis comes. And I think the key change here was that it allows us to look at that quality. It allows us, of course, to obtain different multiple cell types, so we can do some disease modeling and validation. And we have also shown that it's compatible with sequence-based profiling. It does not dramatically change, or it doesn't really change much the molecular profile of the cells. So now we actually are dislocating almost tissue collection from where we process, because we can get tissue from the US now, for instance, or Canada, they can cryopreserve for us and just ship it in the tube. So this is quite a nice protocol, and I'm going to show you some of the details. So this is just a simple comparison of the viability. So if you now take the tissue, half cryopreserved, half not, and then you do it for lung tumor and lung parenchyma, and you look at simple viability by FACS, you see you have a minimal drop of few percent, but it's not even significant. It's about 4% drop in the viability of the tissue, but the viability stays very, very high. You can also do that diseased versus non-diseased, and we have not seen a big problem with cryopreserving COPD tissue. We've tried so far 7 IPF lungs, and they look okay as well. So we think that it will be suitable also for fibrosis. So we could conclude that cryopreservation preserves tissue and cell viability. We also demonstrated that we can then isolate different cell types. So we can now isolate, for instance, fibroblasts. And if you can tweak the protocol, you can cryopreserve your airways separately from your parenchyma. You can cryopreserve your vessels separately. And then you can do things like we did here. You can isolate cells from large airways, small airways, parenchyma versus small airways, tumor or parenchyma, right? So this is just an example of fibroblast on the left top, large and small airway basal cells, and then alveolar cells. It's all published, so I'm going fairly quickly through it, but of course this gives us opportunity for disease modeling. We can now mix and match healthy with disease. And then we also looked whether, for instance, basal cells isolated from cryopreserved tissue will still be stem cells. So we checked all the expression of the markers. We can grow them then in spheroids, and we can also validate that they do still express the markers, and they do also differentiate as we would like them. They still express keratin 5p63 for basal cells, but they also express ciliated cell marker, and they produce mucus. So they still remember that they are stem cells. And that tells us that basal cells from these airways retain their ability to differentiate. We went one step further. We have then taken cryopreserved tissue, sorted EPCAM or enriched EPCAM positive cells, and did single-cell RNA-seq from 3 donors to be able to see whether we preserve all the epithelial cell types we would like to see. And we were very pleased to see that we can, of course, get things like 82s, but we are also getting 81s, which we thought might be fragile if you cryopreserve them, or goblet and clap cells, secretory cells. So we are quite confident that the method preserves, at least on the epithelial level, most of the cell types we are interested in. All right, so I'm going to now go back to the main story. So equipped with that method, we now isolated fibroblasts and 82 cells. It was a pilot project, so the cohort is very small. We had 3 control lungs. We had 3 COPD-1 mild patients because we were very interested to see whether these epigenetic changes will occur early in the COPD development. I don't like to use early because now early has a different meaning in the trajectories, but in mild disease, so to speak. And then we had 5 patients with very well established GOLD 2 to 4 COPD. We matched them by age and by BMI. We tried to match them by smoking history, and they all have been exclusively ex-smokers. And as expected, of course, they varied by the lung function, so we have nice obstruction for the COPD, but the FEV1 for GOLD1 was still quite present. We validated the cell purity of our isolated cells, so we know that they are what they are supposed to do, and then we started looking at the DNA methylation. So essentially, from now on, I'm going to use the word DMR, which is differentially methylated region. So essentially, we are looking at regions which display changes in these little lollipops, which are single metal groups on CpG sites. And then, of course, it can be in either direction. That's what we were after. And this is how DMR would look like. So if you now compare our non-COPD donors versus our severe COPD donor, you see one single DMR, and you see that you have methylation on y-axis. These are individual CpG sites on x-axis. And hopefully you can agree this is this pink shaded region, which doesn't show very nicely on the screen, that there is quite a difference between these 2 types of samples. That's what our algorithm would identify as a DMR. And this is now example from the fibroblast. So this is just one, but we have identified a bit more than 6,000 of those. And then what we did, we then asked, okay, if we look at these regions and now extract them independently from the COPD-1 patients, where would they be? How would they look like? And if you put them all together, that's what you get. So you get this very nice heat map. And I will try to walk you through again. So these are now donors, blue controls, mild in light green, severe in dark green. Each of these individual bars is one region, like I showed you before. So one DMR, you see all the 6,000 of them. And the patients are ordered by the FEV1 declining, so by severity of the disease, if you want. And what you can appreciate, red means higher methylation, blue means lower methylation, is that we have regions in the genome which lose methylation. That's what you see in this first cluster. You also see regions gaining methylation, which is you see in this middle cluster. But you definitely can see that the mild patients or the COPD-1 patients already show the changes that we see later on as well. So they are somewhere in the middle. And that was our very big kind of first observation, that we already see this widespread methylation changes occurring early in COPD1. And this is, of course, the promise, because that may allow us later on to see what are the very first processes which go wrong before the tissue is really destroyed in the severe disease. So that's what we are after. And now you can, of course, do that for other cells. So as I said, we did for 82s, we did also for basal cells. So I'm going to show you only 82s. So now this is the heat map from 82s. We had more changes, and the changes are more dramatic. Then in fibroblasts, we had about 20,000 regions which are changed, or 20,000 DMRs. You see again that we have some gain of methylation, but actually 2/3 is losing methylation, which means we're going to make now the chromatin more permissive for expression, or more decondensed if you want long term. But what was very intriguing, and this is again just 3 patients, so I'm not going to draw any big conclusions, but if you look at the mild ones, now the situation is completely different. We have one which is molecularly looking completely healthy, one which is molecularly looking completely sick, and one which is completely in the middle. And this was very puzzling. As I said, it's only 3 patients, so I'm not going to make a big deal out of it. But if you look at the FEV1, it's absolutely identical. Okay, and that's of course one of the directions we are after now, because we now know where to look. We don't have to go genome-wise. We can now take this 20,000 listen, or even minimize it to 500, and go much larger in a larger cohort and try to see, can we now pick up that some of these patients will decline faster than the others? And that, of course, would be a very nice kind of biomarker if you want in the future. But at the moment, it's just a curious observation. But that tells us that this type of profiling has quite high resolution and might in the future provide us some kind of a sensitive biomarker. Of course, you have to design this study completely differently, make the cohorts completely different as well. This is just a proof of principle idea. Now you may ask, what are the processes which are affected by DNA methylation? And this is just an example from the fibroblast. So if you just link the differentially methylated regions to the next gene and do simple enrichment, what you start getting back is cellular response to hypoxia, focal adhesion, regulation of TGF-beta, and regulation of cell division, which is quite interesting because these are exactly the phenotypes we know go wrong in COPD and fibroblast. So we are not marking random processes. We are picking up what we know is wrong in the disease cell. That tells us that there is some kind of epigenetic marking of key COPD-associated processes. So what we did afterwards, and this was done in collaboration with BI, is we collected 110 candidates, genes from all the profiling that we have done in the fibroblast. We also had RNA-seq, which I'm not showing you here, so we matched integrated that. And we have selected 110 candidates, 50 of them being epigenetic factors. And what we did is probably what you all do, because we were interested in repair function of the fibroblast. We looked what happens if we now knock down these genes and link them to the proliferation capacity of the fibroblast, or link them to the differentiation ability to myofibroblast upon TGF-beta stimulations, the classical FMT assay. And to cut long story short, and again, it's published so you can have a look at it, we had a very high hit rate, probably explaining that epigenetic players are really key in some of these processes. So we had more than 90 candidates that were positive. And of course, many of them have not been linked to COPD. So we now have a lot of work to do trying to understand what they do in the lung and also how they link to the pathology. Just one example of a screen. So that would be an example where we stimulated fibroblasts with TGF-beta, and we looked at alpha-smooth muscle actin as a readout by IF, and we did it both in normal and COPD primary HLFs, human fibroblast. What you can see here is basically just a readout for all the 100 genes. And what you can pick up in red RSA control. So this is what you get if you knock down the genes which are directly involved in the TGF-beta. We picked up in green the 2 epigenetic regulators which have already been linked to alpha-smooth muscle regulation previously. And then the orange ones are basically our new epigenetic regulators which have not been linked to the process before. So that's what we are now very actively pursuing, trying to understand what they do. And just to give you some examples, so these are, for instance, CHAF1A alpha and beta are chromatin assembly factors, which are very important for loading histones during replication. And they have both very strong effects in our assay. And what was interesting is that they actually show some disease specificity. So normal cells react to the knockdown differently than the disease cells. And this was, of course, interesting. If you want to look more, it's published. So I'm going fairly quickly through that part to show you a bit more of the data. All right, so coming back to 82s, what else can you do with type of data? So of course, we now have a metallome. So what else can we do? We can look where in the genome these changes are. And even though there are no good reference maps, there are big epigenetic consortia which did reference maps for the organ. So what you can look is the chromatin marks, you can look at ENCODE data, and what they have done, they profiled different epigenetic regulations across the genome, across different organ cell types to derive functional annotation. So based on epigenetic signatures, you can predict if something is a promoter, enhancer, and what function it may potentially have. So if you overlay now our data with these chromatin maps, what you will see quite clearly is that we have quite a nice enrichment of our differentially methylated regions, either in the regions flanking directly transcriptional start site, so right at the promoter, or in yellow at the enhancer regions. And that is, of course, very interesting because that tells us that, again, they are not randomly distributed. Not only they mark COPD processes, but they actually seem to be enriched in regulatory regions. So if you have to pick a regulatory region in the genome, it would be either a promoter or enhancer. And that, of course, tells us that they might impact gene expression. So the next thing we did matched RNA-seq from the same cells. We did the whole genome dissociated sequencing, and sure enough, we have quite big dysregulation between severe COPD and non-COPD, which we already see partially. So 300 genes were already actually down or upregulated in COPD1. And what you can do is you can ask now, are these changes correlated? Textbook would tell you if you have a methylation in promoter, you're going to downregulate expression. If you remove a methylation from a promoter, you may upregulate it. So you expect in the promoter region, at least, an anti-regulation of most of the genes. That's what the classical kind of view on methylation is. So that's what we did. We look at the Spearman correlation between expression and methylation within promoters, so to speak, or plus minus a couple of kbs from the transcriptional start site. And that's what you get. So if you now use non-differentially expressed genes in blue, you basically have just a normal distribution of these correlations. So nothing really interesting is happening. If you do it for our differentially expressed genes, you hopefully see two peaks. One which is at the negative correlation. That's exactly what you would expect. So that tells us they are anti-correlated, high methylation, low expression, low methylation, high expression. And I'm getting close myself here, so I'm going to just show you this instead. Hopefully that's what I told to my bioinformatician, because he showed me all these nice peaks, but I said, I want to see it. And if you look at it, this is now a heat map. So the heat map on the methylation, which you see is exactly the same as you saw before, with the exception, we are not looking at 534 regions. These are exactly the ones which we correlated in the promoter. Okay, and hopefully what you can see now, the patients are arranged exactly the same. So gene expression is now showing you exactly correlation between a region of methylation gene expression. So look at the bars. Hopefully the pointer is not great, but I can actually walk at least to the people in the room. So this region in the disease, you see hopefully that it's mirror image. So whenever you get red in methylation, you get blue in expression. And this is, of course, a beautiful example of the genes which we think would be regulated, at least classically, by DNA methylation. So this was very nice to see. And that tells us that DNA methylation, at least at these promoters, might drive aberrant gene expression in COPD. So the next thing we did, we said, okay, if we now take all these 500 genes, what are the processes which are regulated? And this is where the surprise came, because I expected nice cellular differentiation, aberrant differentiation of epithelials as something, you know, I could understand as a more kind of epithelial or structural person. What we got back is very much immune cell related or screaming to the immune cells. So our top enriched pathway was type 1 interferon signaling pathway. We see a little bit of chemotaxis and still adhesion, which is quite nice. We do see, if we go down with the process, we see a little bit of differentiation as well. But the main important message was that we see an enrichment in the interferon signaling pathway of these genes. And that was interesting because we started digging in the literature and then pick up on the two papers which drew our attention, where interferons were linked to stemness and repair of 82 cells. And there is a growing evidence showing that if you have high interferons, your 82 cells will not repair properly, they won't grow. So that's what we think is happening, but of course we have to dig later. So we think that upregulation of interferon in COPD 82s is one of the reasons why they don't repair or they don't kind of proliferate properly. But what we were after is, of course, the regulation. Therefore, we asked the question, is this interferon signaling truly regulated by DNA demethylation? So what can you do? You can do a brood assay. So you pick the genes you're interested in, you take your cells, or your cell line, you put ASA, which is basically a DNA demethylation agent, but it's a very kind of unspecific one, right? So you're going to globally demethylate a lot of things. And you can have a look if the genes you're after are now gaining expression or not to get some kind of confidence that they might be epigenetic regulated. And sure enough, that's what we did. So if you compare DMSO versus a 3-day treatment of ASA, you see that in orange that most of the genes which we identify in our interferon signature are now going up. That's what we would predict because they lost methylation. But again, I mentioned this is not a very direct assay, so that could be also very indirect. So we did a bit of a kind of a network analysis to identify what proteins or what factors might be driving these signatures. And we came up to this red shown the transcription factor IRF9. And what was interesting, if you look at IRF9 now within our samples, is that by itself, IRF9 seems to be regulated by methylation. So this graph now shows you correlation of methylation at the specific site in IRF9 and expression across all our cells and disease. And hopefully what you can see is that as the patients become more severe, so to speak, or as the disease stage we are looking at is more severe, then you start having more expression and less methylation. So you can see that. So controls blue have high methylation, less expression. Severe patients now have more expression and less methylation. So that was quite an interesting observation because it seemed to also correlate somehow with disease severity. Is it epigenetic regulation? It's very nice. These mirror images are screaming for it, but it's still an association. And that's where most of the studies end. People associate something and say it's regulated by methylation. By all means, it's not enough because it could be completely indirect, right? So we went a step further and we are very happy we finally got it working. So we wanted actually to functionally study that and we see causal relationship between methylation and expression. So what we've been after and we're doing in collaboration is to develop epigenetic editing tools. So we basically hijack the Cas9 nuclease from the CRISPR system. And this is very nice because you can target Cas9 very specifically to any region you want through gRNAs. So this is cool. The only tweak we do is that we make it catalytically inactive. So it can target, but it cannot cut. And now we fuse to it what you see in this little Pac-Man. We fuse an epigenetic editing domain, DNA demethylase, DNA methyltransferase, histone modifying enzyme, whatever you want. And that allows us to bring that activity to a specific region, change the epigenetic stage and see what happens. So that's the principle. So the question, very focused question here was, can we trigger expression of IRF9 by removing methylation from the region we found in the patients? So that's what we did. And that's where it gets a bit more complicated, but I try to explain you. So what you see here is IRF9 gene on top. And the yellow boxes are exons. TSS shows you where the transcriptional start site is. So this is where the gene will start kind of being expressed. On the left, on top, in yellow, in orange boxes, you will see our promoter probe, so to speak. So that's where we will detect DNA methylation. And you see another one in purple, which is in the gene body. Green box shows our region in 82COPD, which we found to be differentially methylated. And this is where the surprise came as well, because it was not really in the promoter, it was somewhere in the gene body, and we didn't know what to do with it. So we said, okay, let's do an experiment. So we designed two mixes, one which targeted the green region, specifically more into the gene body, and one which targeted the promoter, because we wanted to learn a bit more about the mechanism. And then we targeted our tools, and so what happens? So that's what happens. And I think I need to move to explain you. So if you look at the DNA methylation here, what you see gray is our control. So these are the cells which have seen the tools, but they don't have gRNA, so they cannot target. And this is how the background methylation would be. So it's quite highly methylated, both in the promoter and in the gene body. Oh, sorry, the pointer is not great. So I move, at least for the people in the room. So you see, this is the promoter probes. These are the gene body probes. And you see there's high methylation under background state. If we put now the yellow mix, which is targeting specifically promoter, hopefully you can see that we get a nice loss of methylation in the promoter, the yellow bar going down, but it's not happening in the gene body. Can you see that in the gene body, the bar is still staying, not change much, but it very nicely demethylates here. And the opposite happens if we now target the gRNA mix to gene body. We have a very nice demethylation in the gene body, but not in the promoter. And if we target a mix of both, which means now we mix all these gRNAs, we can demethylate both the gene body and the promoter. So this is actually very cool because that shows us that we have that regional specificity of demethylation that we wanted to prove. So essentially our tools are working. We can deliver demethylation exactly precisely within the region where we want it. Biologically, of course, what does that mean? And this is where the expression came in. So we, of course, took these samples and looked for expression of IRF9. And hopefully you can see again in gray, these are our background cells, control cells, basal expression of IRF9. Actually, those cells don't express much. What if you target the promoter and demethylate the promoter, you get a twofold upregulation of the gene. But if you target the gene body, you have a very nice and robust upregulation of the gene. And this is, of course, very nice because that tells us the region we identified in COPD, even though it's not in the promoter, is a regulatory region, most likely an intronic enhancer somewhere or kind of a cis element in the gene. Right, so hopefully this is just an isolated example to show you what we do and how we think. But hopefully it shows you how kind of nice it is because you go completely unbiased, you follow a process, and then you can find, of course, regions which are quite interested. So yes, we can now do that. The question is, what does that mean for the interference? So we upregulated IRF9, which we predicted to be the master regulator of the interferon signaling. So we, of course, looked in these cells at all the downstream genes that we see upregulated in COPD as well. And hopefully you can appreciate that they exactly follow the same pattern as IRF9. So that tells us we didn't touch them. We did not demethylate any of them with our construct. We only target IRF9. But targeting IRF9 and upregulating IRF9 is actually enough to upregulate the interferon signaling and get us the signature we see in the patients. And what was, I think, even more remarkable, or at least not fully expected, is that really almost those dependent mirrors the IRF9. So if you look at the purple bar, where we get the most IRF9, we also get the most response from the downstream genes. So this is as far as we got so far. But that tells us, of course, that targeted demethylation of IRF9 activates interferon signaling. So that's it more or less. I showed you down a little bit of a summary and I think one or two more slides. So hopefully I was able to convince you that even with a very kind of pilot study in a small cohort, we can say that the methylation landscape seems to be quite changed in COPD in these isolated cells. That epigenetic changes occur, I still keep early, I should remove early, occur already in COPD1. Stage of the disease, and that means also for the future. Of course, the DNA methylation can be used as a biomarker. Not in this setup, but once we know which regions are changed, we could of course change the way we do it. We could go into much more targeted approach, maybe a targeted array or something based on these regions, and then start looking into other materials as well. What was interesting for us mechanistically is that methylation changes occur in regulatory regions and seem to mark some of the key COPD pathways that we see change in the disease, and that hopefully you see that this integrated and biased profiling is a powerful approach for identification of actually not only epigenetic regulated pathways, but also new regulators of the process you are interested in, as long as you have a screening down the road where you can screen for function. All right, so if you want to read more, these are the QR codes. So the first one is the cryopreservation, as I mentioned to Ali already, but to anyone else as well. If you want to share, try the protocol, please do. We love to share it. We love for everyone to use it. We've been actually sharing it already. So we have now collaborated with people doing brain, kidney, pancreas. So it's even transferable across organs. And it was really a huge game changer for us because now we have 107 samples now for COPD, very well characterized, which are kind of there to go back and do validation, for instance, which we couldn't do before. Middle one is our fibroblast story, and then 82 is unpublished yet still, but it's on bioRxiv, so most of the data which I showed you can get there if you want to see details, and hopefully to come soon. And yes, as Gisli kindly mentioned, from June on, officially, I'm not teaching anymore. I'm not having much admin. I can do research stuff. I still cannot believe that it's quite nice. And of course, we are moving. So FLF is now meant to kind of bridge a little bit towards lung fibrosis and try to understand the repair there and maybe compare actually, or try to understand, what decisions the cell make. And this is new ground for me. I mean, I did read a little, but I've never worked with lung fibrosis before. So I would very much appreciate any input. And we are looking for new collaborations. So I have enough space now to actually start things on the side. So if anything we do is interesting for you, I want to try something out, please do reach out. A lot of people involved. So the project started even before I came to Cardiff. So we had, of course, quite a good collaboration with biomedics and with BI. And then Maria and Uwe in pink are the first ones who actually started sorting cells and did the initial analysis. And then on top is now my current group in Cardiff. Deborah, the orange person, is actually a master's student who set up the epigenetic editing in the lab. I can't still believe it, within 6 months. Working day and night, but unbelievably talented student. So this is really amazing what you can sometimes do with master students if you want to try something on the side. And then other collaborators, of course, tissue providers, but also our collaborators at DKFZ, EMBL, and then building slowly also collaboration, hopefully more in the UK. I have anything else? No, I think that's it. Sorry if it was too fast. I went to get excited and go fast.
Thank you. That was amazing. So, is there any questions online? You can text it in the chat. Any questions?
Yeah, thanks very much. Really nice talk. I want to ask about the stability of the methylation status for primary cells in culture. And if you're passaging these cells, does it remain stable through multiple passage or not?
Very little research done on it, to be honest. I'm aware only of one paper in the context of radiation-induced fibrosis when they tried to passage and look at DNA methylation. Their conclusion was that it's fairly stable. It is a worry we had because our fibroblast went through culture. 82s have never seen culture, so 82s are fact sorted and right into the methylation. And I don't have a good answer yet. What we tried to do is we assumed that even if that should not be disease specific, so we treated both in parallel the same way. So we are very strict with how, they were all collected at passage 3 as early as we could to get them pure. Because passage 1 and 2 were still not pure enough for this type of analysis. So passage 3 was the first passage where we had enough purity that we were confident to do that. I think there must be something because I just don't know if it's methylation or actually expression. I would rather question expression because what we see, if you look at 82s, the correlation between methylation and expression is much stronger. If you look at fibroblasts, there is some, but it's weaker. So we also have some regions where we have beautiful methylation, but no change on expression. And in my head, these are probably those which still remember from the tissue what they were. But because we grow them in all this nice cocktail where they can do anything they want, they probably forgot transcriptionally what that means. So my guess would be methylation would be more stable than gene expression. But yes, we are aware that it might be something we have to look at. And of course, again, coming back when you validate, ideally you would like both. So you would like to have cells which were not grown, and then also cells which were grown because our models will always be grown. Right? So question is what is the better system? But yes, so we always try, and also for lung fibrosis, if we can, we try not to grow them. But you are as limited as how you can pull out cells.
So your guide experiments, sort of editing the methylation status was fascinating. When you mixed the promoter and the body, you get a reduction.
Yes.
Why was that?
Yeah, you have a very good eye. Yeah, was it here? Yes. So I think I don't have a good answer yet because we are trying now to do a bit more kinetic and also single gRNAs, try to dissect what is doing what we. So keep in mind it's a mix. We have 5 gRNAs. We don't know what's the individual efficiency of targeting. So I simply at the moment think the mix is simply reflecting dilution of the gene body ones because we keep the same concentration. So if we mix, it means we have now half of the promoter, half of the gene body. And because gene body is much stronger, because that's why it comes in the middle. So that's the most simple explanation I can have, but we don't understand the kinetics yet.
Specifically, I mean, your next slide there shows that you've got a great effect on the mirror of pretty much every interferon-related gene. What about?
Not every. So again, the good question. These are the ones which we saw change in the patients.
Okay.
So we looked for them specifically to mimic whether we have the signature. What we are doing now is we are trying to look at unrelated genes.
Okay, yeah. That's the question. Yeah. So it's just the same thing happening.
We don't know yet. So we are just looking at interferon is very difficult because it's one of these networks where having IRF9 in the system may be just enough to boost the entire network. So it may have nothing to do with specificity of the gRNA. But what we will try to do is, of course, look a little bit at other regions. We did check for guide specificity. The other genes don't come out as secondary targets. But we are in a very kind of new ground here, so we still have a lot of work to do here. We are very surprised it works.
And how specific is it to sort of COPD-related AT2 cells, or is it just a generic? Do you shove a virus on an AT2 cell, is this going to happen?
Well, COVID, the other interferon paper, which came actually from COVID, so the same has been noticed in patients with impetus. There is some evidence after influenza as well. So there's the other paper which I cited. And I was actually laughing because I presented this at an Epigenetic conference in Rome last year. And the person after me was a lung cancer person who showed exactly the same, but the opposite. So they said they actually now try to play with the interferon to make the AT2s not metastasize or not to grow too much. So they were also looking at interferon, but they were actually, we would love to probably suppress it so that the AT2s go up, right? They try to boost it so that the AT2s don't proliferate too much. So I think it will be a fairly conserved property of the cells. But we don't even know. You could also think about STING-CDAS pathway. So I don't even know if this is classical interferon antiviral response, or is it just detecting some kind of double strand, some DNA or mitochondrial changes. We don't know that because we see it only with the signature. What we don't see, and this was interesting, we don't see an obvious dysregulation of the receptors nor ligands themselves. So we pick this up from the downstream signatures of the gene that the pathway is changed. But we try to pinpoint, for instance, if it's alpha or gamma, because we couldn't, when we thought we were going to look at the receptors or ligands, we didn't see anything of this. It's a small sample size as well, so we might just expect statistics. But I think this has been shown already in a couple of studies. So my guess would be this is actually, and interferons have been shown generally to be anti-proliferative in other systems as well.
Not so sure about COPD, certainly for lung cancer proliferation, but fibrosis does it matter how proliferative your AT2 cells are?
So, I don't know, you're absolutely right. So I think for us, we came from the other opposite. We have a cell which doesn't want to proliferate too much, right? So we try to now link it and we bring it. Of course, it's a speculation. We haven't proven what we need is an experiment where we grow AT2s from COPD, where we grow healthy ones, treat with interferons or block interferons and actually control it.
Are there any other functional?
Well, you could look at differentiation, obviously, but we haven't yet. It's quite a complex experiment because, first of all, we have not a great success with growing COPD AT2s. I think Luis, you can second that. It's not that easy to grow them in spheres, and also getting them in the first place is more difficult. So we have a higher dropout rate. So it is something we will look into the future, but we haven't got to do this experiment yet with only cells. We also need a lot of cells. So we can sort about 20,000 cells from 4 grams of tissue. To do culture experiments, we would need to do through quite a lot of, so it's very difficult to do this experiment. So we need a better model system, but I think people are doing it in the mouse. So we could do it with the mouse, or we could do it generally with epithelial cells to prove that they are influenced. Ideally, what you would like to is to maybe also treat with the demethylation, like you would combine it. I don't know.
Yeah, is that? Yeah.
So thank you. I mean, I really like your methods and your tools. One of the things you were talking about is your cryopreserved biobank that you're building.
Yes.
And at the very start, you talked about how the epigenetic kind of controls cellular memory.
Mm-hmm.
So how much history do you take on your samples that you're cryopreserving and storing for future studies? Do you have a full kind of exposure history for controls and disease?
So I must say we had to learn a lot. It took us a long time, probably 1.5 years collecting them to even realize what we want, because when we started it, we had no idea. We get them, most of them are from cancer surgeries. We didn't even know what the type of cancer we influence, whether the cancer will influence the epigenome for some kind of, secondary effect of the distal tissue. Now we are quite optimistic that it doesn't, because our metalomes are completely different than the cancer metalomes. We don't have typical signatures you would see in cancer. So they might be some minor changes, but not dramatic. Because when we collected this, we even matched the cancer type. So we really tried to match as much as we could. That's also why we had so few samples at the end, because, if you try to match everything, smoking would be super important, because it's already shown that smoking causes methylation, even quitting. So they are actually methylation-based algorithms to predict when patients stopped smoking, because there are some sites which revert very fast, some which stay very longer. So you have to be careful there. Some people reported steroid use. So we typically get basic information like I showed you. We do emphysema score index on them to know how much emphysema they have, because we try to, also, you saw even though the data are clean, there are some different patterns between the patients. Could be normal variability. It could be that we just don't have enough information about those patients. We record the medication and any secondary disease they have, so we know the hypertension, diabetes. If they had any other surgery, they would not get any radiotherapy nor chemotherapy yet for the cancer. So that's what we had as exclusion. Infection, we had as exclusion. They try to be as, but we don't know about evolution. We don't know about, exactly where they lived and what they've been exposed to.
Can I follow up on that question? So looking at it from a different way, again, this is speculative. Is it possible to define a person's exposure by their epigenetic signature? So, I mean, we've heard about epigenetic aging, and that's all the rage, and it's crazy people on social media measuring their 20s by epigenetic age. But can you measure their exposure? So, the smoking and pollution and that.
I mean, that's one of the interests we have. So we really need to do a little bit better the basic characterization. So that's what we are setting up as some different models of exposure and looking at kinetics of things and where they are. At the moment, it's very hard to do because the technologies which have been done, either it's association study, and usually have one time points. So you don't know what you are matching that exposure to, right? So it's so you would need ideally even maybe longitudinal, and you have very few data which were done in the culture model. I think that's where you need to come a little bit better. You have to review, basically expose something, see what happens, see the kinetic of it, and see how different that will be between different exposures. What I can tell you is that we see a lot of things which people also see in the smoking. So we do see the smoking signature, but they're all smokers and they're all very heavy smokers. So that seems to be amplified even more, but they probably will be some others. With this sample size, it's very difficult to draw.
One of the real knowledge gaps, and I don't know how we're going to become it in the next 40 years, is understanding what people have been exposed to, how long, just generally, sort of, especially in fibrosis. Again, I know less about COPD, but in fibrosis, the exposure history is often so long ago, people haven't got a hope of remembering certainly what they're exposed to, certainly, what the PM2.5 level of any of our houses were 30 years ago. No one's got it.
I mean, essentially, theoretically, let's put theoretically, all you need to do is to have a large sample size and enough power to do those associations and test them, right? So theoretically, this is very possible. The problem coming back to epigenetic aging, there's going to be a lot of disappointment because there are a lot of new data coming out. And this is unfortunately where the whole field is a little bit going wrong. Because you use bulk tissue or blood, what you are mostly seeing is change in cellular composition of your tissue and not in your epigenetic profile. And I can't even say it loud enough. I show you one slide to show that. So I just went to a meeting. I organized clinical epigenetic meetings in June, a yearly one, and there was two beautiful talks about epigenetic clocks and aging. And one of them essentially taking all published data and showing that if you do cell type deconvolution, all that signature is due to a specific T cell type, regulatory T cell type, which is surviving in the blood for a given number of years. So we have to be careful how I think it's still a very good biomarker. You can still do your age or whatever you want. I think you only have to be careful with the interpretation, because it doesn't actually say you change the epigenome of anything. You just now change the composition of the sample, and methylation, because it's so specific, is going to reflect it. So what is now being done is people need to build clocks based on individual cell types. And I think that will be much more predictive of aging and so on. Having said that, what was quite nice is there was someone showing this. And so there's a big lab in Altos lab in Cambridge. We are doing this, and they showed different types of cells. For instance, pluripotent stem cells, or if you reprogram cells, they actually almost lose their epigenetic aging. But the cells they came from, they are much older, so that you can do a little bit of it. With exposures, theoretically, you should be able to do that. I think we just need more information, bigger labs, and very well characterized samples. I think this is where coming back to your point, I'm completely with you. I think a lost opportunity we have is not even to get the tissue, but to get enough information well annotated that once we have the data will tell us if there is something or not. Because even here, even coming back to fibrosis, Ali, we had this discussion about different types of ILDs. In principle, I don't care. I can just collect them all, profile them all, look at the data and tell the data, can you group yourself into something, right? Or do any type of analysis, and the data will tell me is there a distinction and where the distinction would be.
Three different. It's always three different. But in terms of exposure, it doesn't matter what the disease is. If you're looking for presumably a signature of exposure, then just as much lung tissue as you can get, regardless of what the pathology is, because the exposure should be presumably distinct from the.
So almost like something like UK Biobank, or any kind of population study which is correlative enough. But I think this is difficult because people forget what they've been exposed to. What you could do, and I'm actually trying to do that. We have in Cardiff a wonderful pathologist who can estimate what your tissue has been exposed to chemically. So he can actually run the analysis of tissue of the patient and tell you they've been exposed to this, they have that concentration of this, and so on. So that would be getting it a bit more quantitative.
And then Richard.
Very, very nice talk. I really like it. But for example, for COPD, is it variants that can affect the methylation independently of exposure, or is it exposure that will accelerate the methylation? Is it variant, no? Is this the proximate? You manage to work it out? If we create the variance,
so
we accelerate the methylation.
And of course, one aspect which I didn't mention at all and completely forgot, which will be very important in IPF, is of course the genetic background as well, right? So we in a way we have that information because we do whole genome dissolution, we can convolute it back to the genetic information. As long as we have coverage, we can look at the genes, right? Versus cis versus distal. I didn't show you one thing, which is super interesting in the data as well. And if you look at the enhancers, or if you look at distal elements, we pick up wind. It's screaming with wind. And this is very interesting because somehow promoters are screaming with interferon, why wind is where we see distal. Distal is a bit more difficult to pinpoint because we don't have good reference maps. So what we are trying to do now, and this will take some time, is trying to do our own, because essentially what you need to do now to precisely identify these enhancers, look at the enhancer histone marks, but in your given cell type and in your given disease, not in a organ, and that's the difficulty. If you want to map that very precisely, you actually need to do it in your own data set. That's probably why we are not so advanced. But again, we could go the other way around. We could just identify the most interesting region, the most interesting wind enhancer, change methylation, see what happens. So you could go the other way around, right? First go functional and see if it's important.
There can be a compensation between the lack of methylation. There are elements like the macro RNAs that try to dump.
Absolutely. So one thing we do not understand as well, I say grossly promoters. If you look at them, most of them actually are like in the first half an kb from transcriptional start site. So it's not even upstream where you would expect. It would be in the very first either exon or intronic region. And those often are also places where you would have microRNAs and so on, right? Or in the UTR, like at the end of the gene, in the untranslated regions from the mRNA. So there's definitely much more regulation, we understand. Like, as I said, we are very rudimentary still. I think people are getting much better at the bloods. So blood is probably 15 years ahead. So they really mapped up chromatin structure, histone marks. So you can start doing these very interesting correlations, and they can now map hijacked enhancer in leukemias because they all have this data. We have nothing. I mean, this is the very first high resolution metallome for purified cells here. So we have to do a lot of background work first before we can draw this. But yes, that would be interesting because this is where the promise comes as well. Enhancers, of course, are potentially regions where you can regulate the entire network if you hit it right. And coming back to clinic, something which has used to be fantasy, this epigenetic editing which I'm showing to you, after CRISPR has been approved last year, there is a very first epigenetic editor which is now in clinical trials for hepatocellular carcinoma. They are trying to switch off MYC. Not through methylation, through more of a transcriptional repression, but very similar to these tools. Instead of CRISPR, they have zinc finger nucleases. And actually, there is already a company which has in the pipeline alpha anti-1 deficiency. So they are already targeting it to basically shut down or epigenetically shut down the wrong allele and substitute with the right allele. So I don't think this will be completely science fiction in a couple of years. Just a matter of safety and specificity. And of course, promises, you have the specificity, you can hit a gene, and you are hitting a regulatory layer of that gene. And if everything goes wrong, goes right, and your cell remembers and picks this up, and principally it could be hit and run, so you don't have to take it over life. Of course, I'm idealizing now, but a year ago, 2 years ago, people would completely think this is crazy. And now the progress that has been made, people have put those things in mice already in vivo. So this is really amazing that people already can do with this type of tools.
Yeah, just that immune cells specifically in your samples, just on the basis of this, some of the pathways that kill the regular skin analysis.
No. And like when we started, we actually started sorting macrophages as well. So we had alveolar, interstitial, and monocytes, and we still have them in the freezer somewhere. We never profiled them. But what is interesting, and you're absolutely right to see it in NT2s, but what we have done is we have done a single cell RNA-seq of epithelial cells from the same donors, and we see a lot of immune signaling. So that's probably the next logical step. It's just that, again, from my perspective, at least I need someone who is an immunology person who can guide a little bit, because, this data is only as good as the tissue you get, characterization you have, and how you pull your cells. If I take a decision and I pull them wrong, or I activate them, or I change their fate or something, this data will be trash at the end. So, this is why my confidence zone is a bit less, because I'm not an immunologist. And I think it's much more complex to be able to pull out exactly what you need in each disease. But that's potential, future work, because that's definitely what could be done and then checked.
Anything online? Well, thank you very much. It's perfect timing, just before 4. So once again, thank you so much for coming and sharing your work. And, yeah, we look forward to seeing how it progresses.